Okay, we begin chapter 15 on the immune system, which is the body's defense against infection. Okay, so this is how we're able to live in a world that's filled with pathogens that are on us, in us, and around us all the time, yet still remain relatively healthy most of the time. We'll talk about the two different systems of immunity and explain their separate and interdependent roles. Talk about the relationship between microbial structure and innate immune mechanisms, ID features of the innate immune system that affect acute and chronic inflammation, and explain how activating innate immunity influences disease symptoms. In this section, we'll distinguish between innate and adaptive immunity, describe the different cells and organs that constitute the immune system, and talk about the white blood cell count differential and its role in diagnosing infectious disease. So, essentially, we live in a world that's really pretty dangerous, right? You know from what you've done in lab and what you've learned so far in the course that almost everywhere you look, there are bacteria, and among those bacteria are those that are harmful to human health, and they're called pathogens. Okay, um, it's in the air we breathe, it's in the water we drink, it's on the food we eat, it's on our skin, it lines our digestive, urinary, reproductive, and respiratory tracts. Okay, um, it's virtually impossible to escape. Yet we go through most of our lives without suffering infection and disease most of the time. All right. So how does this happen? Well, we really have three lines of defense between you and the outside world, which is rich with pathogens, right? We have our skin and our mucous membranes and their secretions. That is a physical, chemical, and a biological barrier, okay? The skin, as you'll learn in A&P, has several layers to it. The top layer is called the epithelial layer and is made of simple squamous epithelial cells that are constantly made and shed, and that cuts down on the ability of the pathogen to penetrate into the deeper layers and maybe even get into the bloodstream. We sweat and we produce oils, and that tends to flush the skin of those pathogens, and it creates an environment in which very few microorganisms like to grow because it makes what's called an acid mantle, meaning that the sodium content is very high and the pH is very low, and that retards the growth of a large number of organisms. Okay? And then beneath the epithelial layer is the dermal layer, which is connective tissue filled with white blood cells, which stand as a guard against those pathogens getting into the blood supply, which is the last thing really that we want to happen. Okay? You have a similar setup in the mucous membranes that line the reproductive, the urinary, the respiratory, and the digestive tracts. Okay? The mucus serves as a way to trap pathogens and flush them out of the system, and the epithelial layer that lines the tract is constantly made and shed. And underneath that epithelial layer, we're going to have um, a dermal layer that has immune cells in it. Okay, and that tends to keep us free of infection. Okay, same thing we see in the urinary tract, the reproductive tract, and the respiratory system. Okay, in fact, in the respiratory system, the epithelial tissue is modified so that it contains cilia, which can move the secreted mucus with the pathogens and the particulates up towards the oral cavity, or we can either spit out the material or we can swallow it. And when that encounters the acid secretions of the stomach, that kills even more microorganisms. Okay, so we've got quite a few um, defenses, first lines of defense against infection. Okay, now in the event that something penetrates those barriers, okay, We have a class of white blood cells which are called granulocytes, okay? And they're named after the dyes that stain those cells so that we can visualize them under a microscope. So if you take a drop of blood and you smear it on a microscope slide and then you stain it with right stain, you'll see different classes of granulocytes, the eosinophils, the basophils, and the neutrophils. Okay, the eosinophils get their name from the fact that they take up the red eosin in the right stain, and the granules stain red, and the granules of eosinophils contain antihistamines and antiparasitics. Okay, the basophils stain with the methylene blue in the right stain, and their granules contain histamine and heparin. And then the neutrophils have granules that stain both with the eosin and with the methylene blue, so their granules look pink, and they contain antimicrobials. Okay, these chemical weapons are crude, okay, and they sometimes hurt healthy tissue, but they do a pretty good job of cutting down on whatever leaks through our first line of defense. Okay, the last barrier between the pathogen and the deeper tissues in the body is our adaptive immunity, and that's made up of our lymphocytes and macrophages. And what they do is they mount an attack against any pathogen that leaks through the first two lines of defense by generating cells that recognize foreign antigens, bind to them, and then destroy anything those antigens are attached to. Okay? And there's two arms to adaptive immunity. There's cell-mediated immunity, whose destructive abilities come from a population of cells called cytotoxic T cells. And then there's humoral immunity, who use basically B cells that mature into what are called plasma cells, which secrete antibodies, which then bind to antigen and target anything that they're bound to for destruction through a variety of mechanisms. Okay? And because these different layers of protection use different tools to keep us healthy, it's very rare that something gets through all the protective layers and makes us sick. Okay? The white blood cells of innate immunity are shown here, the polymorphonuclear leukocytes, the monocytes, the macrophages, the dendritic, and the mast cells. Okay? We'll go through what each of these do. All right, this is what they would look like in an EM micrograph, where the colors have sort of been added after the fact. Okay, scanning EM here, and you can see that the red blood cells are pretty simple. They have thick rims and a hollow, sort of a concave center. They're basically filled with hemoglobin and an enzyme called carbonic anhydrase, and their main job is to carry oxygen and some CO2 around the body. Okay, but the white blood cells have all the cellular machinery, nucleus, organelles, everything, so they have the ability to reproduce, repair, and grow. Okay, where the red blood cell lacks that. Okay. The neutrophils are granulocytes with pink staining granules and nuclei that are between 2 and 3 lobes. Okay, you got little constrictions and lobes there. That's where the chromatin is in the cell. And their main job is to destroy microbes through phagocytosis, as well as the release of antimicrobials in their granules, which induce a process called apoptosis in the target, which is programmed cell death. Okay? They are the most abundant of the leukocytes. Okay? You can remember the relative abundance of the leukocytes using the mnemonic "Never let monkeys eat bananas," where neutrophils are the most abundant leukocyte, lymphocytes are the second most abundant, eosinophils follow that, and then basophils follow that. Okay? And then, never let monkeys. After leukocytes comes monocytes, which become macrophages, and then eosinophils, and then basophils. Okay. The white blood cells can be grouped as granulocytes and agranulocytes. The granulocytes, which are the neutrophils, basophils, and eosinophils, have visibly staining granules in their cytoplasm, whereas the agranulocytes do not. That doesn't mean they don't have granules, it just means that the granules don't stain. And the significance of the granules is that they're chemical weapons used to destroy pathogens in a variety of different ways. Okay? Basophils and eosinophils have different roles. Okay? A basophil has blue staining granules that obscure the nucleus and contain histamine and heparin. And the job of the histamine is to promote inflammation, while the job of the heparin is to slow the clotting process long enough for the other leukocytes to come in and do the job of destroying any pathogen that gets in through a wound. Okay? Eosinophils turn off inflammation because their red staining granules that pick up the eosin in the dye contain antiparasitics and antihistamines. Okay, so the idea is that once we've beaten back the invader using the granulocyte action, the eosinophils kind of come in and calm it down, and their other job is to paralyze and destroy um, parasites such as protists or uh, um, worms, okay, and also to engulf and destroy antigen antibody complexes. Okay, so the time when you see an eosinophil count goes up, it's when you have a parasitic infection or when you're perhaps suffering from an allergic response. Okay, neutrophils tend to kick up when you have a bacterial infection, and basophils tend to kick up when you have inflammation. Okay, mast cells contain granules that have histamine and heparin. So mast cells are sort of like fixed basophils. They're kind of locked into the tissue they're found in. And their job is basically the same as the basophil, right? When they get the proper cue from tissue damage, they release the histamine and the heparin. The histamine sets off the inflammatory process, which we'll see a little bit about in a second. And the heparin maintains a liquid environment that doesn't clot quite so quick, so that the neutrophils and the the um, the macrophages can come in and basically eat up any of the garbage, right? Eat up the pathogen, eat up dead tissue, and so on. Okay. Monocytes have a round nucleus that takes up almost, well, it, no, sorry, the nucleus is more kidney shaped. Okay, and it basically turns into a macrophage. And the job of a macrophage is to work with the lymphocytes in order to promote our adaptive immunity. So what the monocyte does is it differentiates into the macrophage. Differentiate just means to change over time. Some of them also become dendritic cells, which are like fixed macrophages. And what they do is they sit there, and when an invader comes in, they eat him through endocytosis, and then that fuses with a lysosome, and the enzymes in the lysosome chew up whatever it is the macrophage took in, chop it into little pieces, and then the macrophage presents those little pieces on its membrane surface along with a membrane protein called MHC2. And that collection of molecules on the surface is like a warning to the helper T cells and the cytotoxic T cells that there's trouble in town. And those T cells that recognize that antigen presentation are the ones that are stimulated to divide and then to go out and destroy infected tissue. Okay, the B cells kind of do the same thing. The B cell encounters the antigen that it's supposed to recognize, and that's the first step of it getting clicked on. And then when it encounters an activated T cell that has seen the same antigen, it finishes the job of getting clicked on, and then it becomes a plasma cell that squirts out antibody into the bloodstream, and that antibody eventually travels to wherever the antigen is found, binds to it, and marks it for destruction. So the antibody's job basically is to attack antigen that's kind of out in the open and not inside our own tissues. The dendritic cells are like fixed macrophages, and what they do is they eat up invaders and present them to the T cells, doing a job similar to what the macrophages do. And a lot of them are concentrated in places like the spleen and the lymph nodes, which are, as we're going to see, found primarily in the neck, under the arms, along the respiratory and digestive system, and in the inguinal region, and in the cervical region, right? I already said the neck. And the reason they're concentrated there is because that's where they can drain areas of the body that have openings to the outside world, okay? And by taking that tissue fluid and filtering out the enemies, and then sending that tissue fluid eventually back into the bloodstream, we filter out the invaders, and we also warn the white blood cells that there could be an attack in the offing, okay? So let's look at phagocytosis to get an idea how it works.
Several types of cells in the immune system, neutrophils, macrophages, and dendritic cells, can engulf microbes by a process called phagocytosis. Here a bacterium binds to the surface of a phagocytic cell. The phagocyte extends pseudopods that engulf the bacterium. The bacterium becomes trapped inside a vesicle called a phagosome inside the cell. A lysosome, which is a vesicle filled with digestive enzymes, fuses with the phagosome. Inside the resulting phagolysosome, enzymes destroy various components of the microbe, and ultimately, the microbe itself. The cell releases the microbial debris outside the cell.
Okay, so sometimes when you're sick and you go to the doctor, one of the ways that he can determine what might be wrong with you is to do something called a white blood cell differential, which is part of a broader test called a complete blood count. Okay, and what he's looking at here are just the total number of cells in a drop of blood, and he wants to break each cell down by its class, and then see if the expected percentages match the percentages that they find when they actually count your cells. Okay, and if you've got an elevated or a depressed count of any of these classes of white blood cells, that can indicate what type of infection or what type of disease process you might be undergoing. Okay, the same really is true of looking at the red blood cells as well, right? If the red blood cells are abnormally low in number, abnormally high, or abnormally shaped, that can indicate disease as well. Okay, so you literally put these things on a slide, you stain them, and then you sit there and you count them. Okay, and this can be done by a human, or this can be done by a computer. Okay, so we ID the neutrophils, eosinophils, basophils, lymphocytes, and monocytes. Right? Remember my mnemonic, right? Never let monkeys eat bananas. Right? So the basophils, the least abundant. The neutrophils, the most abundant of the white blood cells, right? The other cells that are in there are going to be your red blood cells, your erythrocytes, and your platelets, which are called thrombocytes, okay? So you can see normal counts, okay? And then you can see what happens when there's a bacterial infection, when there's a viral infection, when there's a parasitic infection, okay? And when there's an allergic response, all right? So you can see what goes up, what comes down, okay? The eosinophils, it should also be shown on here as increased during an allergic response as well, okay? The elevation of eosinophils is called eosinophilia, okay? And it can be either a normal response to an infection, or it can be result of abnormal red bone marrow, okay, where the process that produces the eosinophils gets misregulated, and it becomes sort of like white blood cell cancer, a little bit like leukemia. Okay, you can see what happens with the viral infections, right? You reduce your neutrophil count, you can increase your lymphocyte count, and your monocyte count will increase under certain infection types, right? So this is a diagnostic tool, okay? Basically, we look here and then we ask the question, what's off? Are there are there cells that normally wouldn't be there in great abundance, there in high abundance or in abnormally low abundance? And what does that mean for the possible disease process? Okay. Um, One place where the white count gets elevated, but there is no infection going on, is leukemia. Okay. And the problem there is that the bone marrow, the red bone marrow, has suffered either a viral infection or a mutation of some kind, and the stem cells that produce the leukocytes begin to produce large numbers of white blood cells that really don't do anything for the host. And the result of that is that even though the white count is really high, their susceptibility to infection is also elevated. Okay, this can be inherited. This can be acquired. Okay, and the types of leukemias are generally based on the cell type that's produced in abnormal numbers. Okay, let's take a look at the basics of the lymphatic system.
Lymphatic vessels recycle excess tissue fluid that accumulates in the interstitium as a result of normal capillary leakage. The walls of the lymphatic vessels act as one-way valves, only letting fluid enter. Once in the vessels, the lymph is advanced toward the systemic circulation. Along the way, the lymph is filtered through several lymph nodes where it is inspected for foreign substances. Upon reaching the thoracic duct, the lymph re-enters circulation.
Okay, so when we talk about the lymphatic system, we've talked about the cell players, right? We've talked about the leukocytes, but we should also talk about some of the organs, okay? And they include the tonsils, the mucosa-associated lymphatic tissue, which is found primarily in the mucosa of the respiratory, the digestive, the urinary, and reproductive and respiratory system, okay? The spleen, the thymus, okay? And we could also throw in there the liver to an extent, okay? And essentially, the B cells and the T cells get their name from where their ancestor cells come from. B cells come from bone marrow every day, okay? Your red bone marrow, which is primarily in your axial skeleton. So that would be your skull and your ribs and your vertebral column, your scapula, the proximal ends of your humerus and your femur, your coxal bones, your sacrum, your coccyx, okay, and your sternum. When you give red bone marrow for tests, okay, like if you volunteer to give red bone marrow, they pay you lots of money, between $1,500 and $2,000 for them to stick a needle in your hip. They basically give you a hip pointer and withdraw some of that red marrow for study. And it's usually a pretty big needle, okay? And it's kind of painful. So it's the sort of thing, like, if you can deal with the pain, it's probably worth the money. But if you're kind of a wimp, maybe you want to stay away from that, okay? The T cells begin production in the bone marrow, but finish production in the thymus, which is a temporary endocrine organ that sits in front of the heart and behind the sternum and shrinks as we get older, okay? The T cells are selected within the thymus for immunocompetence, so they have to have the ability to both recognize foreign antigen and also tolerate our own antigens. And if they flunk either of those tests, they are either destroyed or they're rendered in a state of what's called anergy, where the cells are alive, but they're not actively participating in immunity. Okay, and then what they do once that happens, once that selection's all done with, is that they move out from the thymus and then they populate the areas of the body where they're going to do their work, such as the lymph nodes and the mucosa-associated lymphatic tissue, the spleen, um, and also take up residence in the bloodstream at various locations. Okay, so they go out, they get deployed. Okay, the reason that this is an effective way of guarding against invaders, all right, is because once the process is over, the B cells and the T cells that you've made have the ability to recognize between a billion and a trillion different antigens other than your own antigens. Okay? And that's usually enough to keep you healthy for most of your life. Okay? I don't know if you ever looked at the number 1 billion or 1 trillion, but it's a really huge number. Okay? And the reason that this is possible is because the genes that contribute to the production of the antibodies and the production of the T cell receptors that are going to do the job of recognizing these antigens are actually produced in the course of this selection. Okay, so what happens is that in the nucleus of the cells that are eventually going to be the lymphocytes, right, we undergo DNA rearrangements in the antibody genes and in the T cell receptor genes until we produce a working gene that generates a functional receptor or antibody, which is going to be unique from any other white blood cell that produces a T cell receptor or an antibody. Okay? It's kind of like playing poker, right? With a deck of 52 cards, you can get 5 cards out of the deck. How many different possible hands could you have? And that ends up being 52 times 51 times 50 times 49 times 48. Okay? That's a lot of hands, possible hands, right? Well, the same is true with the production of B cells and T cells, except that the quote-unquote deck of cards is hundreds of segments, different segments of DNA that can be combined in a variety of ways, and in the course of that recombination, we can remove or add nucleotides. And so once that process is finished, you get to an awful lot of possible antibody and T cell receptor genes. And the reason we have to do it this way is because if we just dedicated the DNA in the nucleus to doing nothing but making a billion different antibodies or T cell receptors, that would basically use up all your DNA, and you wouldn't have any DNA left over for anything else. Okay, so that's the process. Okay, so while everybody's adaptive immunity is functionally identical, okay, the parts that go into it are different. Okay, just like an insect wing and a bird wing do the same job, but are made of different stuff. Okay. Secondary lymphoid organs are the lymph nodes, the spleen, the Peyer's patches, the malt, the gulp, tonsils, appendix. Okay, and those are areas where basically the T cells migrate to once they've been produced, and they're usually accompanied by macrophages in order to do the job of screening for pathogens effectively. And there are usually some granulocytes in there as well. Okay, and they just populate those organs and do the job of screening whatever comes past them, right, in order to pick off the pathogen, generate large numbers of white blood cells that recognize that pathogen, and then they can go out and do the work of destroying it wherever it's located, right? The B cells produce the antibodies, so that's the humoral immunity. The T cells modulate the adaptive immunity through a special cell called the helper T cell, all right? And then the cytotoxic T cells do what's called cell-mediated immunity, where they actually seek out and destroy infected tissue in the body, okay? So the B cells are primarily attacking loose toxin or loose pathogen, right? While the cytotoxics are going after infected cells, okay? All right, so if we look at these different locations of the immune organs, you can see them here, right? We've already talked about most of these. But notice where most of the lymph nodes are, right? You got them, a lot of them in your neck, a lot of them in your armpit, a lot of them along the respiratory and digestive system, a lot of them down by the reproductive and urinary system. So we got concentration of what we call inguinal lymph nodes, axillary lymph nodes, cervical lymph nodes, thoracic and abdominal pelvic lymph nodes, okay? And the reason for the location of these lymph nodes is because they're all near systems that are open to the outside world, right? The digestive system is open to the outside world at two ends, right? The respiratory system at one end, the urinary and reproductive system at one end, okay? And you could even consider the breasts as open to the outside world at one end, okay? And so all of that, it means that the lymph nodes need to drain large amounts of fluid that potentially contains pathogens and then return that eventually to the blood at what's known as the subclavian vein. Okay, the spleen does a job similar to the lymph nodes. The lymph nodes work on a liquid called lymph, which is the leftover fluid that isn't reabsorbed by capillary beds at the venule end. Okay, and that ends up going into these lymphatic vessels and eventually to the lymph nodes. The spleen is basically doing the filtering work with blood. Okay, so blood goes into the spleen. We look for bad guys. We gobble them up. We warn the white blood cells that there's an infection, and then we send that filtered blood back out into the body in order to do the work. Right. The tonsils guard the opening to the respiratory and digestive system, and macrophages and lymphocytes live there. The Peyer's patches and the mucosa-associated lymphatic tissue are collections of white blood cells just below the inner lining of those systems that stands guard for pathogens that are on our food and drink that could get through and into the blood. And of course, the red bone marrow is where we make blood, okay? And that's found primarily in the axial skeleton and the proximal humerus and femur, okay? So that's where everything's located. Okay? We'll talk about the physical barriers to infection, talk about salt, galt, and M cells in innate immunity, and chemical barriers used by cells of innate immunity. So we talked a little bit about skin already, right? Keratinocytes make up the bulk of the epithelial layer of the skin, which is the upper layer, and it is basically set up so that as keratinocytes are made, they migrate to the surface, and then they are shed, and then they are replaced by younger keratinocytes. So we're constantly shedding our skin and leaving a trail everywhere we go, and the importance of that is that we don't give bacteria time to colonize, right? Because we don't want that to happen, okay? So no colonization means no infection. The skin oils, which are made by the sebaceous glands and the sudiferous glands, which make the sweat. We already talked about how they flush the surface and create an environment that is hostile to bacterial reproduction. And then we also create an environment that is friendly to bacteria that are good for us. Okay, an example on the skin would be Staph epidermis and Staph aureus, right? Staph aureus is pathogenic because it creates enzymes that can drill through the skin and sometimes get into the blood, and that's what a staph infection is all about. And then Staph epidermis doesn't do that, right? And so when Staph epidermis predominates on the skin, it bogarts all the space, and the result then is that we stay relatively free of skin infections, okay? So that's our own microbiome kind of helping us out, okay? The skin-associated lymphoid tissue, or the SALT, right, recognizes the microbes that can slip past the physical barrier. Langerhans cells, which are dendritics, can eat up the microbes before they get into the skin and also warn the immune system that you have a potential infection, right? And the cells that recognize that will be prompted to reproduce, okay? Other barriers, okay, mucous membranes we've talked about, and we talked about what mucus does. There's also enzymes like lysozyme that weakens bacterial cell walls, and lactoperoxidase, which basically creates harmful oxygen species that could damage the pathogen. Microbe-associated molecular patterns are basically patterns of peptidoglycan and lipopolysaccharide that are recognized by our immune cells regardless of what kind of pathogen that they're on. Okay, so this is a little bit like, it's almost an antigen, but not quite. Okay, and so our innate immunity can sometimes pick up on that and attack it. Okay, we've talked about MALT. Okay, gut-associated lymphoid tissue is basically the mucosa-associated lymphoid tissue of the alimentary canal, the digestive system. Okay, Peyer's patches are MALT that is found in the area where the small and large intestine join up, which is called the ileum. Okay, and the reason that the Peyer's patches are located there is because it's so close to the colon, which of course is packed with bacteria, the most abundant being something called E. coli, with which we have a symbiotic relationship. The E. coli bacteria eat our waste products and produce, in turn, B vitamins and vitamin K, which are important for our metabolism. M cells are specialized cells that take up microbes from the intestine as well. Okay. Our lungs have an upper respiratory that's lined with pseudostratified ciliated columnar epithelium, and goblet cells in that epithelium produce the mucus, which is then moved up towards the oral cavity by the action of the cilia, right? Sneezing expels irritants from the respiratory tract. The mucociliary escalator is just the description of that pseudostratified ciliated columnar epithelium doing the job of moving that mucus up towards the oral cavity. Okay, organisms that make it to alveoli aren't easily destroyed, but eventually wandering macrophages will find and kill them. They ingest and kill bacteria and send out chemical signals to attract other cells of innate and adaptive immunity. And then, of course, what happens once that stuff's destroyed is it becomes part of the lymphatic drainage from the lungs and is filtered by the lymph nodes that are found in the thoracic region. Okay. The acidic pH of the stomach is a sterilant, which destroys most bacteria. It's hydrochloric acid that's produced by the stomach. Okay. Enzymes like lysozyme in our saliva, tears, and our sweat weakens bacterial cell walls and gram positives. Remember, those are going to stain purple with the gram stain. Superoxide radicals generated by our host enzymes kill pathogens such as lactoperoxidase, which produces free radicals that hurt pathogens. Okay, remember that free radicals are electron poor species that steal electrons from healthy molecules and destroy them. Defensins are positively charged peptides made by a lot of different cells like skin, lung, and genitourinary and GI tract, and they destroy microbial cytoplasmic membranes. They're effective against both gram-positive and gram-negative bacteria, fungi, and viruses. Okay, and so the way that these are going to work is by essentially punching holes in the membrane and causing the cytoplasm to leak out. Okay? We'll talk about inflammation and its process, and talk about vasoactive factors and cytokines, and acute and chronic inflammation. So let's begin with inflammation.
Although many things can trigger inflammation, our focus here is on how microbes cause the response. The process begins with the infection itself. Microorganisms trigger inflammation when they're introduced into the body and begin to grow and produce compounds that damage host cells. Resident immune cells, called macrophages, wander into the infected area and engulf and digest the invading organisms. They help to clean up the infection and, in the process, release inflammatory mediators, chemicals that call for more help. Some of these chemicals increase vascular permeability and stimulate vasodilation. Others are small proteins called cytokines that are made and secreted by cells as a way to communicate with other cells. Here they diffuse into the vasculature and stimulate endothelial cells to express specific receptors called selectins. Selectin binds to carbohydrates on the surface of neutrophils. snagging the cells as they flow by in the bloodstream and slowing them down, such that they roll along the endothelium. Inflammatory signals trigger these rolling neutrophils to express adhesion molecules called integrins on their surface. The integrins lock onto adhesion molecules called ICAM1 and VCAM1 on endothelial cells. The tight adhesion stops the rolling, and the neutrophils begin to stretch out along the endothelial surface. Damaged tissue cells in the area of inflammation release bradykinin, a 9-amino acid polypeptide that helps loosen the tight junctions between endothelial cells. Neutrophils can now initiate extravasation, in which they squeeze through the loosened endothelial wall and into the tissues, where they can help macrophages attack the invading microbes. Bradykinin molecules also bind to other cells of the immune system, called mast cells, that are in the area, causing them to release histamine. The histamine further loosens the endothelial cell junctions, allowing more fluid and cells to move out of the capillaries. Bradykinin, when it attaches to capillary cells, induces the cells to synthesize prostaglandins. Prostaglandins stimulate nerve endings, causing pain, which draws awareness to the infected area. The 5 cardinal signs of inflammation are redness, warmth, pain, swelling, and altered function at the affected site.
Okay, so with chronic inflammation, anytime you see the word chronic, that means over a long period. So a degenerative process that takes place over a span of time. Okay, persistence of a foreign entity in the body can cause permanent tissue damage resulting from the inflammation. Examples include infection by Mycobacterium tuberculosis, which is the pathogen that causes TB, or Actinomyces bovis. Okay, granulomas are just the body's attempt to wall off the irritant, and this causes stimulation of the inflammatory response, and that results in tissue damage. Okay, because inflammation is always going to result in damaging some tissue that is relatively healthy. Okay. Non-living irritants include foreign bodies like wood splinters, asbestos, and surgical implants that can produce chronic inflammation. Okay. Other sources of chronic inflammation include repetitive motion injuries, okay, such as tennis elbow or carpal tunnel syndrome, okay, where repeated movement irritates and inflames tissue around nerves, causing pain. Okay. We'll talk about how phagocytes recognize foreign cells, explain how phagocytes kill engulfed cells, and list ways that pathogens avoid phagocytosis, and talk about how our body stops inflammation. Okay. So, to recognize an alien invader, right, we have to have a T cell receptor and an antibody that can bind to it effectively and tag it for destruction. Okay. The antibody, once it finds the antigen that it latches onto, can then allow a process called opsonization to take place, where the antibody, by surrounding and binding to the antigen and then working with something called complement, makes it easier for endocytosis by macrophages to take place, and then you can destroy the pathogen relatively easily. Okay. The other way that antibodies can work is through a process called immunoprecipitation, or we sometimes call this agglutination, right? And the idea here is that we trap the pathogen or the antigen in a big meshwork of antibody and antigen, and it basically comes out of solution and becomes immobilized and much easier to destroy. The other way that the antibodies do their job is through the activation of something called complement, which is a series of serum proteins made by the liver that works along with antibodies in order to help destroy anything the antigen is attached to. And we'll see exactly how that happens when we get to the area of complement. Okay. The other way it does it is by cloaking viral proteins that allow viruses to dock and invade host cells, or cloaking toxins so that they can't produce their toxic effect. Okay. And in all of these cases, once the antibody surrounds it, it makes it a candidate for destruction by macrophages. Okay. So those are some of the mechanisms that antibodies used. Okay, these are the B cells that become plasma cells that do this, and so we call this humoral immunity because it's the body fluids that mediate the delivery of the antibody. And um, in the old days, when we didn't understand anything about health, I think I talked about this earlier in the course, we used to think that the balance of the body's fluids was responsible for your health, and that we called those the humors, right? Phlegm, blood, um, urine, um, mucus, sweat, and so on, right? And so back then, the cure for everything was to bleed you, right? Okay, so um, phagosomes are basically membrane enclosed vesicles that contain the solid material that was pulled in by the phagocytic cell, in which case here we could be talking about a neutrophil or an eosinophil or a macrophage. Okay, what will happen is that the lysosome will then fuse with it, and it has very powerful proteolytic enzymes as well as enzymes that can break down cell wall components and can punch holes in the cell membrane. So basically what happens is that this collection of chemical weapons inside the lysosome comes in and wrecks the ingested pathogen. Okay. Oxygen-dependent mechanisms include oxygen radicals like superoxide and hydrogen peroxide, again, which tear away electrons from healthy molecules and wreck their ability to do their job. Macrophages, mast cells, and neutrophils also produce nitrogen intermediates like nitric oxide and nitrite and nitrate ions, which have a similar effect on their targets. They are basically free radicals. Okay, so very damaging species. Sometimes we run into a pathogen that can slip past the defenses. Okay, phagocytosis is very effective at killing microbes, but some pathogens have evolved ways to basically slide by this type of attack. Some bacterial intracellular pathogens like Coxiella have evolved to live within the toxic phagolysosome so they can deal with that environment. Others like Shigella and Listeria escape from the phagosome before a fusion can take place, and then they live in the cytoplasm of the macrophage. And then Salmonella remains in the phagosome but secretes proteins that prevent fusion with the lysosome. Others, like Shigella, enter a host cell and kill it by triggering a host process known as apoptosis. So they kind of become a suicide pill. Death of the macrophage saves the bacterium from phagocytosis. The apoptosis is a normal process in which the cell kills itself in such a way that limits damage to surrounding cells. It's important during development, right, when we change from an embryo to a fetus to a neonate. But in this case, what we're doing is we're using this trick against the cell, right? Normally, cells don't go into apoptosis until they've lost so much information in their nucleus that they can't utilize the genes that maintain their function anymore, and they just say, the heck with it, we're checking out, okay? And then they get replaced by new cells, right? We'll talk about host-specific antimicrobial functions of interferons and how natural killers recognize and kill infected host cells, and talk about toll-like and nod-like receptors and how they work, okay? So back in the 80s, right, interferons were thought to be a wonder drug because they were shown in the lab to be effective against cancer and against viral infection. Okay, and so there were certain drug companies that made large amounts of these interferon-based drugs in an attempt to market them as a cure-all. Well, it turned out that a lot of these compounds were hepatotoxic, meaning that they damaged the liver before they were able to exert their beneficial effects on the cancer cells or on the virally infected cells. So they were pulled from the market. So there was a reason that when the body uses interferon, it uses it in limited quantity. Okay. Um, Basically, the interferon is a way for the virally infected cell to warn its neighbor cells that there's a virus in the area, so it can produce enzymes that will destroy viral intermediates that are produced when the viruses try to reproduce themselves. Okay. Cytokines are another way that we can warn neighboring cells of danger. They're produced by eukaryotes. Intracellular infection triggers their release, and then we start generating enzymes that defend against viral replication. Okay. Examples include type 1, which are high antiviral potency interferons, including IFN alpha, beta, and omega, and then type 2, which are immunomodulatory, which include IFN gamma. Okay. Type 1 interferons bind specific receptors on uninfected hosts and induce the production of double-stranded activated endoribonucleases. An example is a protein called dicer. Okay. Protein kinases are also induced, which phosphorylate and activate E1F2, which is required to translate viral RNA. Okay. Type 2 interferons activate white blood cells, such as macrophages and natural killers and Ts, and increase the number of MHC antigens on the surface of the infected cell and make it easier for that cell to be recognized and destroyed. Okay, so this is an MHC called MHC1, and if MHC1 on an infected cell is holding up an alien antigen, that's basically a signal to the immune system to destroy that cell. Okay. Natural killer cells ID and destroy infected cells by degranulating and releasing chemical weapons that induce apoptosis and leakage of cytoplasm from the target by punching holes in the membrane. They recognize the MHC with the unusual antigen. Okay. But if there are no MHC molecules, the cell will be destroyed by perforin. Okay, so if we have a goofy antigen or if we have no complex, then something weird is going on and we need to wreck that cell. Okay, the only way the cell escapes destruction by a natural killer is to present a self antigen and an MHC1, right? And then the cell will see that and say, you're fine, and we can let you go about your business. Okay, so the white blood cell leaves that cell alone. Antibody receptors include antibody-dependent cell-mediated cytotoxicity. Okay, and essentially this is a way, again, to utilize the antibody to destroy the infected target. Okay, so some examples here, right? Natural killer cells recognize cells that have lost expression of MHC class 1. The cells store cytotoxic molecules like perforin and lysosomes, and recognition of an infected cell triggers release of these molecules, so that's called degranulation. And then they kill the target by causing the cytoplasm to leak out. Right? So this basically punches holes in the membrane. The perforin makes a pore in the target and destroys the integrity of the membrane, and then cytotoxic proteases called granzymes are also kicked out from the natural killer. They pass through these pores, enter the target, and tear it up. Okay, so punch holes in it, start it leaking, and then send in destructive enzymes, and then it's toast. Okay, and so this is part of a process called immune surveillance, which wrecks cancer cells when they form on a daily basis. Everybody in this class gets cancer 2 or 3 times a day, but the reason you're not dying of cancer is because of this immune surveillance, so that when a cell goes rogue, it gets destroyed before it gets a chance to spread. Okay, and that's why people with suppressed immune systems are more prone to what are called opportunistic cancer. Okay, the faster the body can detect the presence of pathogens, the more quickly it can begin to deal with them. The more quickly it deals with them, the better the outcome of the infection, but it takes time for adaptive immunity to do its work. All that time, the pathogen can grow and cause disease, but bacteria and viruses have unique structures that immediately tag them as being unusual. These include peptidoglycan, flagellin, single or double-stranded RNA, and lipoteichoic acids, which we don't have in our own tissues, okay, unless they've been infected, all right? As we talked about earlier, these structures have what are called microbe-associated molecular patterns, or MAMPs, which can be recognized by toll-like receptors present on various host cells. These toll-like receptors, along with nod-like receptors, are pattern recognition receptors. Once bound to an MAMP, the toll-like receptor sends a signal to the interior of the cell to begin whipping out interferon and inflammatory cytokines, and that speeds up the process of inflammation. These toll-like receptors were first picked up in insects. They're an evolutionarily conserved glycoprotein present in the cell membrane of a lot of eukaryotes. Okay? Humans have numerous toll-like receptors, each of which recognizes different MAMPs on pathogens, making them an innate defense mechanism with a degree of specificity. Okay? So it lies somewhere in between adaptive and innate immunity. Okay? As an example, TLR2 binds lipoabinomannan from mycobacteria, zymosan from yeast, and lipopolysaccharide from spirochetes. TLR4 binds LPS from gram-negatives. Note that these receptors bind fragments of structures after they're released from the microbe. They don't interact with the whole organism. Okay? So they need pieces, broken off pieces. The cytokines made after an MAMP-TLR interaction float away, bind to receptors on cells in the immune system, and direct them to engage the invading microbe in the inflammation process. Okay. As a result, battle-ready levels of cytokines are going to be produced as long as the components of the system are intact. Right? Cytokines can influence the function of cells that are part of innate or adaptive immunity. Although toll-like receptors are restricted to membranes, okay, some cytoplasmic proteins also recognize MAMPs. Nod-like receptor is an example. It was named because of the similarity to a family of plant proteins involved in pathogen resistance, which are sensors of MAMPs. When bound to an MAMP, the nod-like receptors trigger a signaling pathway different from that of toll-like receptors. The result, though, is the same: activation of cytokine genes in the nucleus. Nod-like receptor activation initiates assembly of inflammasomes that proteolytically break down precursor cytokines into active ones. Once made, the cytokines are secreted that bind the target immune system cells and initiate inflammation and adaptive immune mechanisms to respond to the intracellular pathogen. Okay? So basically, it's just a way to kick off immunity quicker and more effectively. Okay? So once that inflammation's going, we bring blood to the area, we elevate the temperature in that region, the region swells up because of the extra fluid, okay, and there's pain. And all of that is designed to begin the process of the destruction of dead tissue and the destruction of the pathogen, and then setting the stage eventually for tissue repair, right? So you can see some of the different MAMPs, their sources, their host cells, and their locations, okay? Next thing we want to talk about is fever and what fever serves. So let's take a look at fever.
Hey guys, if you can't tell by my voice, I've been pretty sick this week, but I was determined to get a video out to you guys nonetheless, and I figured it would only be suitable to answer the question, why do we get fevers? So let's dive into it. A fever can simply be anything different than the average 98.6 degrees that the body is normally at. But since the body can fluctuate a degree or two during the day due to environmental factors, that isn't a very accurate definition. A better way to characterize a fever is by oral temperatures higher than 100 degrees Fahrenheit. But since oral temperatures still aren't incredibly accurate, the best way to measure a temperature is to go through the ear or rectum, and a fever then would clock in at temperatures at 101 degrees Fahrenheit or higher. Fevers are of course most commonly caused by viruses, but they can of course be caused by other things such as toxins in the environment, a messed up hypothalamus, or of course a heat stroke. But as the number one cause of fever today is a virus, that's what we'll be focusing on today. When a virus infects your body, it oftentimes travels through your bloodstream. But thankfully for us, our body has white blood cells. These guys travel around our body, and when they find a harmful bacteria, they produce pyrogens that travel to our brain. These pyrogens then travel to the hypothalamus area in our brain, the area that regulates our body temperature. Our hypothalamus in turn then cranks up our body temperature and gives us the fever. But what's the purpose of making our body feel miserable? Well, most bacteria that lives in our body lives at the nice and cozy 98.6 degrees Fahrenheit that our body normally functions at. So, when the body heats up, the bacteria either stops reproducing or dies, making our body get better over time. But with this information in mind, a controversial topic is brought up. Should we take medication to lower fevers? The answer is no, unless it's a dire situation, because by lowering body temperatures, you're allowing the infection to continue to grow and hurt us. And let's not forget about those nasty side effects. Some side effects of ibuprofen, the active ingredient in most fever-reducing medicines, are, wait for it, chest pains, weakness, upset stomach, diarrhea, rash, dizziness, headache, pain, numbness, bloody stools, shortness of breath, slurred speech, seizures, itching, and chills. So, next time you're taking a sick day off of work, put that medicine away and let your body do its work. Disclaimer, the statements and ideas provided here in this video have not been approved by the FDA, the Food and Drug Administration. These statements are not intended to diagnose, treat, cure, or prevent any disease, so if you're having any problems with your body, be sure to visit a professional healthcare advisor. Thanks for watching. If you enjoyed, be sure to rate, comment, and subscribe. Like I said, this week I've been pretty sick, so sorry for the kind of sick voice and the shorter video, but hopefully next week will be a much better video. Once again, thanks for watching, and if you would like to see some other content by Science Saturdays, be sure to click on the pictures below. Science Saturdays, bringing free education to the modern world.
Okay, the next thing we want to tackle is complement, and we'll talk about the classical and alternative and lectin pathways, and talk about the role of complement in host defense. So let's take a look at complement.
Regardless of whether the complement system is activated by the classical, alternative, or lectin pathway, the activated complement proteins are involved in several important processes associated with host defenses, including inflammation, attraction of neutrophils, opsonization, and cell lysis. Complement C3A participates in inflammation by inducing changes that cause the walls of local blood vessels to become more permeable. This facilitates movement of phagocytic cells from the circulation into the tissues. Complement 5A induces a directed chemotactic migration of phagocytes to the site of complement activation. Complement C3b functions as an opsonin. It binds to microbial cells, making them more susceptible to phagocytosis. Complement proteins C5b, C6, C7, C8, and C9 react together to form a hole in the cell membrane. When C5 convertase converts C5 into C5a and C5b, the C5b binds to the cell surface and combines with C6. C5b6 binds C7, enabling it to insert into the membrane. Binding of C8 is followed by binding of several C9s. The hole generated by formation of this membrane attack complex causes the cell to lyse. The complement system consists of a group of over 30 serum proteins that play an important role in both innate and adaptive immunity. Complement proteins participate in host defense processes such as inflammation, attraction of phagocytes to an infected area, enhanced effectiveness of phagocytes, and lysis of cells. Complement proteins are activated by a cascading mechanism. There are 3 pathways of complement activation: the classical pathway, the alternative pathway, and the lectin pathway. The classical and alternative pathways are demonstrated in this animation. The classical pathway requires interaction of antibodies with an antigen, usually on a cell surface. Following binding of antibodies to cell surface antigens, C1, which is composed of 3 proteins, R, Q, and S, attaches to the constant region, Fc, of adjacent antibodies through its C1q subunit. C1s then acts on C2, cleaving it into C2a and C2b. Then C1s cleaves C4 into C4a and C4b. C2b and C4b bind to the cell near the antigen-antibody complement complex, and C2a and C4a are released. This complex of C4b and C2b, called C3 convertase, has protease activity and cleaves C3 into C3a and C3b. C3b becomes bound to the cell surface, whereas C3a is released. C3b then absorbs to C4b2b to form C4b2b3b. This structure is called C5 convertase. The alternative pathway does not require the presence of antibodies. Activation begins with cleavage of C3 into C3a and C3b, either spontaneously or by a blood protein. C3b binds to foreign substances. Factor B, a blood protein, attaches to C3b and is cleaved into 2 fragments, BA and BB, by factor D, forming C3bBB, analogous to C4b2b. Both structures, C4b2b and C3bBB, are called C3 convertase. C3 convertase rapidly cleaves more C3 into C3a and C3b. C3 convertase is further stabilized by properdine, which allows another addition of C3b, forming C5 convertase.
Okay, so that brings us to the end of Chapter 15. This is just a couple of the videos that we looked at during the course of this podcast. You're free to review them when and where you want. I will see everybody in the next podcast, and I thank you for listening.